chapter 9. Chapter 9 is dealing with like microbial nutrition and growth. So any nutrient that is required for an organism, we'll call it an essential nutrient. And of course, all organisms are going to need carbon, hydrogen, and oxygen, and nitrogen, and phosphorus, and sulfur too. We don't talk about the sulfur as much in this class, but there are plenty of amino acids and other molecules that are going to have sulfur in them that are absolutely integral to the function of our proteins. But we have talked a lot about the CHON and Ps, right? A lot. So we know that those are going to be required. So macronutrients are nutrients that are being needed in large amounts, but we also have micronutrients like our trace elements that you absolutely have to have in your diet, but they're going to be in much, much smaller amounts. They're usually things that are important for the protein function. So this is going to bring us to breaking down our organisms basically by their carbon sources and how they get their energy. All right, so to start with carbon, we have heterotrophs, right? Heterotrophs and autotrophs. Heterotrophs are organisms that must get their carbon in an organic form. So they depend on other life forms to turn it into an organic form, and then you eat those things and get their organic molecules from them. So obviously we're heterotrophs. We don't make our organic molecules. We get them from the food that we eat. Absolutely not are all organisms that way, because plants, for example, will use inorganic carbon dioxide and fix it into organic molecules. So they're able to take that inorganic carbon dioxide and turn it into organic molecules. And then we can eat them to get the organic molecules. Make sense? So autotrophs are able to take inorganic carbon dioxide and automatically turn it into the organic molecules. The heterotrophs will eat a lot of different things to get those organic molecules. So that's our carbon sources. Any organism, we've learned about growth factors a lot. So just to remind you guys that a growth factor is anything that has to be provided to that organism that they can't make for themselves. Like with us with vitamin C, we can't make vitamin C, so we have to have it provided to us in our diet. If you don't, then you get scurvy. So we have to have it provided in our diet. That's our growth factor. It has to be provided in the environment. So we also have energy sources. So we learned about our carbon sources. Those are our heterotrophs and our autotrophs. For our energy sources, you might get your energy from the sun, or you might get your energy from the food that you eat by breaking it down. So there's a lot of energy contained in the bonds that hold our food molecules together. And when we break them down, we can release that energy. That's what chemotrophs do. Chemotrophs break down the chemicals into smaller parts and use the energy from breaking it down to get that energy for their cells. Phototrophs use sun energy to drive their own cellular processes. They convert sunlight energy into cellular energy. So obviously a photoautotroph is something that will be using sunlight to fix inorganic carbon dioxide into organic molecules. So we're starting to see how those things are related to each other. Energy source versus carbon source, they are different things. Phototroph, energy from the sun. Autotroph, carbon from inorganic. Okay, so we are chemoheterotrophs. So we get our energy from the chemistry of the things that we eat, and we get our carbon from all the organic molecules from stuff that we eat. Also, all our stuff comes from the things that we're eating. And once again, photoautotrophs, energy from the sun into cellular metabolism. We're going to use that energy to drive fixing inorganic carbon dioxide into organic molecules. And then chemoheterotrophs, like us, we're using the other molecules that other plants or other organisms made, and then breaking that down to use for both carbon source and energy source. All right, there are also saprobes that are a little bit more specific. These are still technically heterotrophs, so they're like a subset of the heterotrophs. So they are just only going to be breaking down dead or decaying matter in order to get their nutrients and energy. So this is mostly bacteria and fungi. So then we have extracellular digestion for saprobes, where like if you're something that breaks down dead and decaying things, you're a bacteria. Obviously, you can't eat up that whole organism. That's not going to work. So what they do is they secrete enzymes, and those enzymes that they secrete out of their cells will break down that material, and then they can absorb up what they've broken down. Okay, so that is extracellular digestion for those guys. How do we absorb our nutrients? All right, so this is where we're going to start talking about our membranes again. So our membranes are selectively permeable. They are meant to only let certain things cross the actual membrane. And if you want anything that can't naturally cross that membrane to be getting in and out, then you need to have a way to control that through channels and things like that. We're going to talk about that. So to get your stuff into the cell, you're going to need to be able to find a way to allow it to cross, because as it turns out, our membranes being semi-permeable or selectively permeable, those selectively permeable membranes only allow water and certain tiny molecules to cross. So obviously things like your nutrients, like your organic molecules that you're trying to bring into the cell to break down to use to make your DNA and your proteins, that ain't going to cross the membrane on its own. So you're going to have to find a way to get it in if you broke it down outside of the cell, okay? So, or if you're going up to something and you're wrapping your whole cell body around it, like we talk about with phagocytosis, you're eating up a bad guy cell. You need to be able to have a mechanism to be able to do that. And being able to break that down and get it into the cell, we need mechanisms to be able to do that. So this brings us to the first concept, which is diffusion. Diffusion is just movement of molecules with a gradient. If I have a big old column contained by a big old net of bouncy balls, right? And I cut that net open, are the bouncy balls going to stay in that column? No, they're going to go everywhere. That's diffusion. So moving from an area of high concentration to low. Ideally, what physics tells us is that it wants to be even everywhere, okay? That's the idea. This brings us then to osmosis. And osmosis is just the diffusion of water. And water can cross our membrane, whereas things like salt outside of the cell can't. So that's why if you have a cell that's 95% water, and you put it into a very salty solution, let's say 50% salt, it's a lot more salt than it is water, right? So it's high salt outside, low salt inside. The water wants to balance that out. The salt cannot move across. So the water compensates instead. The water can move across. So that's why the water, in very salty situations, the water moves out of the cell. It's trying to water down the salt outside of it to make it equal on both sides. Because it's like tiny bouncy balls that are in that big old column. If I have a whole bunch of tiny ones that can fit through the net, they're going to come out and they're trying to disperse out and spread everything out evenly. That's the idea of the water. The water can move, other things can't. So that's why we have osmosis. Osmosis is just diffusion of water. That's it. Diffusion of water. And it's going to be dependent on the concentration of water. So we are used to thinking about solutions and the concentration of the things being dissolved in the water. Don't forget, it's not just about the concentration of the thing in the water, but also concentration of the water itself. So that's what drives osmosis, is the concentration of the water. All right, and so you guys have probably learned about this with osmosis, hypertonic, hypotonic, and isotonic, right? And so if you put something with a lot of salt in the cell, let's say you have a really salty cell and the water's pure water around you, what's going to happen with the water? It's going to move into the cell this time to try to water down what's in the cell to make it equal on both sides. Because again, the salt can't cross. So those are two examples of how water is going to move. Isotonic conditions, the salt and the water are the same on both sides. So in isotonic conditions, the water concentration, okay, how many water molecules there are inside versus outside is the same, isotonic. Iso means basically equilibrium, okay? In hypotonic conditions, this is where the hypotonic refers to what's outside of our cells typically, okay? So whatever your cell is. So hypotonic is low tonic, so low salt compared to the cell, right? So this would be a situation where if you were a column of bouncy balls, the bouncy balls want to move out, right? So in this case, it's hypotonic, your high salty cell, the water isn't going to be, the salt wants to move out, but it can't. So the water instead moves in to try to equal it out. Okay, so only the water is moving.
It swells.
And it swells, and that can burst the cells. This is a situation where having a gram-positive organism, they are more resistant to these sorts of changes because that wall is going to help maintain that shape a lot more than the thin mess will for the gram-negatives. Okay, so they can handle being in hypotonic and hypertonic conditions better than the gram-negatives can typically. Okay, so hypertonic condition, what's outside of you is hyper, it's high in salt. So hypertonic, referring to that salt environment, more salt, or it could be sugar. Doesn't matter, but anything that could dissolve in water, the thing that's dissolved in it, the solute, is high. It's hypertonic. So there's, let's say we pack you in high levels of sugar even. Again, same thing as salt, right? It's going to draw the water out of the cell because it's trying to dilute out its environment to make it equal on both sides. So you can limit microbial growth with this if you have enough salt or sugar. This is what jam and preserves are about. This is why they have such high sugar content, is because that high sugar content prohibits the growth of microbes through osmotic pressure. Too sugary, it draws the moisture out of the cell and the cell dies by plasmolysis. So here's our picture of these different situations. Isotonic, things are the same concentration inside as out. Okay, so everything's just normal, copacetic. Hypotonic solution, it's more watery outside than it is inside the cell. I've already told you guys, cells are about 95% water. Even if I say that to you guys, if they were 95% water, and if we're to put you in pure water, obviously pure water doesn't have anything in it. So your cell is going to be really struggling to try to make it equal concentration inside and outside. Is it ever going to catch up to the outside? No. Even no matter how much it waters down what's in the cell and it swells and swells and swells by bringing in that water, it's still going to have more salts and things compared to what's outside. So that's why the cells will burst, because they'll eventually just get to that point where it's not good anymore. That's hypotonic. Hypertonic, putting it into salt, for example. Now, to water down our outside environment, we push our water out of the cell because only the water can move. So pushing that water out of the cell to water down our outside environment, and that'll cause our cell inside to shrink down, and that can kill the cell. This is called plasmolysis, right? All right, whoops. I'm trying to make this bar go away. Okay. All right, so that is movement across the membrane, but not everything can cross the membrane. Like I just said, water can cross the membrane. Other non-polar, tiny non-polar molecules might be able to cross the membrane on their own. But most things can't cross the membrane. So what if you need to get things in and out of the membrane? Obviously you do. You need to get nutrients in there. You need to get junk out of the membrane. You can't just rely on things crossing the membrane naturally through diffusion. So there are different methods by which the cell can move things across a membrane. The first one we're talking about is passive transport. These are still reliant on the process of diffusion. So this could be what we talked about with simple diffusion, where our water, our osmosis effect goes. It's just simple diffusion. Simple diffusion of water is osmosis. In facilitated diffusion, now it's like I opened a little door on my net of bouncy balls, and I can open that door and the bouncy balls can move out. The net was containing them and they couldn't move. And now I've basically made a channel for them to be able to go in and out. So facilitated diffusion, we're still relying on diffusion. We're still relying on the gradient to allow transport. So can you move things from high concentration to low concentration? Yes, because it wants to balance out. Diffusion will drive that. However, if you want more nutrients in your cell, let's say you have a pretty high concentration of nutrients, but you want more, and there's not a ton outside, but you want to keep bringing in more, now you're going from an area of low to higher, and that is not going to be driven by diffusion. So that is not what facilitated diffusion will do for us. So then you're going against the gradient, and that's called active transport. So anytime you're moving anything against the gradient, or you're trying to make the movement of diffusion faster than what it naturally would be, that will require energy. So if you're ever pushing anything against the natural force of things, then you're going to need energy to make it happen. And this is active transport. Obviously, with how quickly your body needs to send nerve signals, you can't rely on diffusion to regulate all of your signaling through your potassium and your sodium and your protons and things like this. So with cells signaling each other, that has to be nearly instantaneous. And so you're going to rely on active transport to allow for your cells to reset those nerves so you can send impulse after impulse after impulse, right? So that's going to rely on active transport. Eating up anything outside of the body is going to rely on some sort of active transport. There's all different kinds of active transport. So we have some carrier-mediated active transport, where basically you'll bind to your channel, and then energy will allow you to pass it through the channel, even against the gradient. So energy is the only thing that you'll have to submit some ATP as energy. And basically ATP, remember we have those 3 phosphates linked to each other. They're always pushing against each other. So they're always wanting to kick off one or more phosphate. And when we break that bond, that's what drives our proteins being able to do work. How, you might ask? Well, I'm very glad you asked. We're going to talk about that in chapter 10. All right, so that's what chapter 10 is going to focus on. But ATP will have to be used in all of these active transport examples because that's our cell's energy. So anytime you're endocytosis, which is bulk transport, so this could be the cell swallowing something big up, like we have with phagocytosis, requires energy. And now you're probably still wanting things to go faster than what diffusion could manage, or you're fighting against the gradient, either way. All right. So that is diffusion and osmosis. And we know our osmotic pressure, basically how salty something is in the cell versus outside the cell, and vice versa, can affect our microbes. We just talked about that, right? So what are some other things that can influence the growth of our microbes? Obviously, they might be picky about temperature. The microbes that make you get sick, if we freeze them, are they going to be able to make you sick? No, they can't do anything. Just like if I froze you, you wouldn't be able to do anything. So we could talk about the extremes of the temperature, but some of these guys are a little bit more sensitive, even within the range that they function in. So temperature, gases, a lot of time the biggest gas that can have effect on these guys will just be oxygen, which we take for granted, but many of these guys can die in the presence of oxygen, or they might not care about it at all. We have pH. We already know the importance of pH. I was explaining this to my husband and the things that I've explained to you guys about pH. If your proteins are folding based on hydrophobic and hydrophilic environment and like engaging with the wateriness of the cell and hydrogen bonds that are basically helping that whole protein hold itself together, helping your DNA hold itself together. If you shift that pH, now you're introducing too many or too few hydrogen ions, and you're affecting how the proteins can fold and therefore function, how your DNA is bound together, and therefore how it functions. And so shift in pH can be a very drastic effect on cells. So pH is important. Osmotic pressure, that's our salt versus water concentrations. Radiation exposure, hydrostatic pressure. This is literally like as you go deep into the ocean, the pressure of the water on you. Okay, so actual pressure of the environment or barometric pressure. Moisture levels, and then of course the presence of other organisms. So let's get into some of these guys. Cardinal temperature is the optimum growth temperature. All right, so this is usually a range of temperatures. Most of our microbes will want to live within a certain, between this temperature and this temperature. And then outside of that, it's not as friendly to it, all right? That's the cardinal temperatures. And then we can group microbes based on their cardinal temperature preferences. Here we have the psychrophiles. This is the guys that like the cold. The mesophiles, these guys like about room temperature. Like what we live in is mesophile temperature. We're mesophiles, okay? Thermophiles, the guys that like hot. Then we have the hyper or extreme thermophiles. These guys like really hot, so above boiling even, and so on and so forth, okay? So again, psychrophiles, these guys will grow below 15 degrees Celsius and often can even grow at freezing temperatures. We also have psychrotolerant microbes. These guys will tolerate cold, but they don't prefer it. Okay. Mesophiles, these are the middle temperature guys. So this could be anywhere from 10 to 50 degrees Celsius, but most of them prefer the range of 20 to 40, just like we do. 20 is like room temperature. 40 is like above, basically a fever would be like 42-ish. Okay. So your normal body temperature is 38, 37.6 or something like that. 38, we'll say. Okay. Most human pathogens between 30 to 40, and that makes sense when I tell you that 37 to 38 is the human body temperature. Thermoduric temperature or microbes can withstand high temperatures, even higher than normal. So they can even withstand pasteurization, for example. All right, so thermophiles, these are guys that just prefer to live in high temperatures. So these guys thrive between that 45 to 80. I won't test you guys on the range, by the way. I will expect you to know that thermophiles grow at higher temperatures. Mesophiles grow at the middle range temperatures, okay? And if I give you a range of this temperature to this temperature, I'll usually say room temperature to body temperature, for example, so that you have a reference, okay? I won't make you memorize the numbers. All right, let's move on to the gases. So exposure to certain atmospheric gases. Oxygen is the biggie big. And then presence of how much carbon dioxide might be in an atmosphere can affect these guys as well. So if they're sensitive to the concentration of carbon dioxide in an atmosphere, then that would be another predictor of how well they can survive. We have 3 categories of microbes based on their ability to deal with oxygen. We have the guys that use oxygen and therefore must be able to detoxify. Oxygen is capable of making free radicals, like ozone, for example, and other molecules that it might be able to bind with. And the cell has to be able to deal with those byproducts of oxygen and using oxygen to create our energy. And so if you are an organism that uses oxygen to create energy, which we'll talk about in chapter 10, and it'll make more sense. But if you need oxygen, like we do, if I deprive you of oxygen, you die for sure, 100, right? So microbes can be that way. If they use oxygen, they have to be able to detoxify it. We also fall into that category. So that's the first one up here. The second one, they can neither use it nor detoxify it. So these will be, our examples for these ones might be stuff like Clostridium species, like C. diff. It's an anaerobe. And so what that means is anaero, no oxygen. It can't use oxygen. It doesn't use oxygen. And if you put it in oxygen, it'll kill it. And that's why it has endospores, so that it can hunker down in this horrible toxic oxygen environment until it gets somewhere where there's no more oxygen. All right, yes.
Would there be questions like that on the exam?
If there is, I won't ask you specifically necessarily about Clostridium. If I say there are organisms like Clostridium that are anaerobes. And so we're actually going to be talking about anaerobes and aerobes in a lot of detail in chapter 10. So yes, you're going to be expected to know what those terms are and what they mean and how they apply to the organism.
Just be a description of it and say
Yeah.
either use or toxic.
Yeah, unless it says in your actual study sheet that you have to know that Clostridium is anaerobic, then I won't expect you to know that offhand, if that makes sense. I would put that in the question. Kind of like on the last test where if I did talk about mannitol salts, I told you what it was so that you could apply that. I would expect the same thing. Yeah. So we have organisms that can neither use oxygen nor detoxify it. And then those that do not use oxygen, but they're still able to detoxify it. And we have terms for these guys as well. So like we've said, we have oxygen. It can make things like ozone or singlet oxygen, which is just the O by itself. And that these guys are reactive and can cause damage to the cellular structures. And we actually use this in our lysosome. Remember how I told you about our lysosomes contain digestive enzymes and toxins and things like that to kill bad guys? We actually package stuff like this in our lysosomes to kill bad guys with. That's how toxic it is. We use it as ammunition. So these guys, like singlet oxygen and stuff like that, cause damage through oxidation, which again, yeah, redox reactions. You guys remember that stuff from chemistry? Redox reactions and how electrons are going to be exchanged. We'll talk about that in chapter 10. Yeah. So also superoxide, which is O2 with an extra electron. Remember how hungry those oxygens are for those electrons. They can just steal them. There's some hydrogen peroxide and hydroxyl radicals. If we can have H pluses floating around, we can have OH negatives floating around. So all of these guys, our cells have to be able to deal with this stuff because we engage with oxygen on a regular basis. So we have things like superoxide dismutase, which takes superoxide, which is O2 negative, and turns it into hydrogen peroxide. And I already told you hydrogen peroxide is deadly, so no worries. They also have enzymes that break down hydrogen peroxide into water and oxygen. Okay. So we absolutely are going to be learning more about this in the lab in the second part of the semester. So we're going to be doing tests on our organisms to see if they have catalase and things like that. So if I'm telling you that hydrogen peroxide, there's an enzyme that can break called catalase. Remember ACE enzymes, right? So catalase enzyme breaks down hydrogen peroxide into water and oxygen. Ever wondered why whenever you put hydrogen peroxide on your cuts, you see bubbles? It's oxygen being released. Your body is using catalase to break down hydrogen peroxide into oxygen because your body has catalase. It's one of your body's way to deal with toxic oxygen products. So that's also a reason why if you are working in a typical like hospital environment and you're treating wounds, why we don't advise that you use hydrogen peroxide for deep wounds, because it'll kill your own cells and provide more food for deep tissue wounds if you don't get that hydrogen peroxide really deep down in that wound, right? So home treatment of wounds, if you have like puncture wounds and things like that, this is why we don't If you're using just superficial surface wounds, it's fine for that. Or if you're using it for oral rinses, it's great for that. But if you're getting it down in the tissue and you're not 100% sure that you're really getting down deep into that tissue, you could just be killing off your own cells in addition to causing damage to potential bacteria. But if any of them survive, now they've just got more food and more damaged cells to eat off of. So it can be dangerous really quickly. Okay. All right. Now let's move on to our organisms categorized basically based on how they interact with oxygen. We're going to talk a lot about aerobes and anaerobes. Okay. Aerobe. What does aero suggest to you? Does it suggest oxygen to you? It does to me too. So hang on to that. Okay. So we're going to talk a lot about aerobes and aerobic respiration, especially in the next chapter. Well, chapter 10. Okay. So aerobes are aerobic organisms. These guys need oxygen. Aerobes, oxygen, they need oxygen. Okay. So they also are able to deal with oxygen. Remember how our guys in this previous description up here, those that use oxygen and can detoxify it, there's none that use oxygen and can't. Notice that? If you use oxygen, you have to be able to detoxify it. So that's just the default. Okay. So come back to this. Aerobes. If you are an obligate aerobe, you are obligated to use oxygen and be able to deal with it. All right. Obligated. You have to do it. So obligate aerobes have to have oxygen. You are an obligate aerobe, right? Facultative anaerobe. You could also think of a facultative anaerobe as being a facultative aerobe. Facultative, when you hear that word, it means it can switch between. So facultative anaerobes, they don't require oxygen for their metabolism, but they can use oxygen. And when oxygen is available, it's better. They can make energy more effectively. We'll go into the details of how facultative anaerobes are going to make their energy without oxygen through a process called fermentation. So not a small one, right? We know fermentation is an important one, but that these organisms can rely on fermentation as their sole energy productive means if they need to, but they're also capable of using oxygen. And as it turns out, using oxygen is the absolute #1 best way to make energy, period. So these guys can use oxygen when it's available, but when oxygen is not available, the whole reason whenever you're brewing your beer or your wine or your alcohol of whatever kind, you will remove as much air as possible is because you want to encourage fermentation. And that's what gives you your alcohol as your byproduct. Okay. So that we'll talk about fermentation and how that works in chapter 10. So we've got our aerobes that need oxygen. We've got our facultative anaerobes. They can be without oxygen, but they can use oxygen too. They switch between. We have microaerophiles. These guys just don't need as much oxygen. Microaero, small oxygen. And then our actual anaerobes, pure anaerobes. So there's kind of two categories of our anaerobes. We have our obligate anaerobes, again, obligated to be without oxygen. These are the guys like our Clostridium guys, like C. diff, that they cannot tolerate the oxygen at all. They don't use it and they can't tolerate it. So our obligate anaerobes, ones that have to be without oxygen. These guys cannot even tolerate the oxygen. All right. But we also have another group called the aerotolerant anaerobes. And I hope that that speaks to itself to you guys. So this is the group that doesn't use oxygen. They're without oxygen and aero, right? And they're aerotolerant. So they can be in the presence of oxygen. They just don't use it. So to a certain extent, they can handle detoxifying the oxygen. All right. Are we clear on all those groups? Obligate aerobes, facultative anaerobes can kind of switch between, micro aerobes, less oxygen. And then we have our anaerobes that are obligate and our anaerobes that are aerotolerant. All right, osmotic pressure. Osmophiles. Osmophiles, they are game to live down with the high solutes. So if this is salts, like sodium chloride, chlorine is a halogen, if you know your chemical tables. So chlorine is a halogen, so this is halophiles. So halophiles are literally osmophiles for salt. All right, if you're dealing with sugar, You're not osmophile, you're a different kind of, you're osmophile, but you're not a halophile, right? So a halophile is a type of osmophile. We'll call them sugar files. You don't need to know the words, but they would be a different kind of osmophile, okay? So we have the obligate halophiles versus the facultative halophiles. What do you think those are, right? We're back to those terms again. So in case you can't figure it out, we're going to be using these terms a lot. Obligate halophiles have to live in the presence of salt. If you put them in an area like what we live in, which is kind of mid-range salt concentration, these guys would die. They need high levels of salt. They've adapted their cells to deal with the high salt. So they're not going to do well without the salt. So they have to have high salt. Our facultative halophiles, these guys can live in areas that are normal salt concentration versus high. They can live in kind of both, right? So again, facultative, switch between. All right, next we're going on to our associations between organisms. We're going to focus on the symbiotic side, but there's two sides to this. Symbiotic, where there's close dependent relationship. So we're symbiotic, the organism actually depends on the relationship somehow. At least one of them does. In a non-symbiotic relationship, this is where organisms relate to one another in their environment, but it's not actually required. All right, so you could think of this as basically like a synergistic sort of relationship with you and your dog at home. That is not a symbiotic relationship, right? Your dog can leave and go get food from somewhere else if it wanted to. But the fact that you're giving it food helps it out. That's a synergistic, and they're giving you love and reduced anxiety and all this back. So that is not symbiotic. That is a synergistic. Does that make sense? That dog doesn't have to be in that relationship. If they had to be in that relationship, that would be symbiotic. So we're going to focus on the symbiotic guys. There's three types of symbiotic relationships. If the two organisms are both benefiting, and at least one of them absolutely depends on that relationship, then that's mutualism. Okay, so it's a type of symbiotic relationship called mutualism. Mutualism is a type of symbiosis. In commensalism, the commensal is benefiting, but the other one's just there along for the ride. Okay. Nobody's getting harmed though. Mutualism, both benefit. Commensal, one benefits, the other one don't care, not affected. Then parasitism, one benefits and the other one is harmed because of it. So if you have a tick that's feeding off of you, it's taking away your nutrients, maybe giving you diseases, and it's literally biting into the tissue of your body, causing you damage, that's a parasite. So any organism that has a parasitic relationship, that parasitism, right, that's a parasite. So viruses, when they come into your cell, and they're utilizing your cell's resources and tools, that's parasitism. All right, when you stub your toe and it gives you an open wound and it gets infected with the bacteria from the environment, that's parasitism. You're not benefiting from that. Now, we have microbes in our gut that help break down our nutrients for us that we can't break down on our own. The gut bacteria are doing that for us. So we're getting nutrients from them, but they're also getting a safe environment as well as nutrients from our food. That is mutualism, right? So we clear on the differences between those three biggies? Great. Good, okay, we already did this, so I'm just gonna click through unless I see something I know that I care about. All right, so moving on to biofilms. We've already kind of talked about biofilms and how things will stick to each other from capsules or slime layers. And then based on our ability to read the room, right? Whether we're seeing a lot of waste product, okay, the guys around us must be happy, they're making a lot of waste, or whether we're seeing a lot of products made from things dying around us, which could also signal to you, maybe I should get out of here before I die. That is quorum sensing. So basically sensing the chemical signals in that population. All right, so they're not actively like thinking and talking with each other, but they can respond to that environment within that biofilm and then decide how to react within it based on that. And that's quorum sensing. So we always have our pioneer organism that sticks to a surface. Other things stick with it. The stickiness just allows more stickiness. And it'll continue to grow until we get signs that maybe we shouldn't grow anymore. And we can go and escape and maybe go create new biofilms. All right. Binary fission is how bacteria divide. This is not the same as mitosis. All right, so we don't have the same steps as mitosis, but it is kind of similar. But this, just know that eukaryotes divide their normal cells when we make copies, exact copies. That's mitosis. Bacteria, when they divide, it's called binary fission. And one cell will also always become 2 cells. And so it's exponential growth from there because 1 becomes 2, and then each of those can become 2, right? So 2 becomes 4. Does that sound a lot like that PCR that we talked about? 1 DNA becomes 2, 2 becomes 4, 4 becomes 8, 8, 16, 16, 32, 32, 64. It's the same with this. That's called exponential growth. So binary fission works very much the same way. All right, so we're doubling that population at every step, just like we doubled our DNA with PCR. In bacteria, they follow a growth curve. The first initial part where we're not having any visible increase in the population, this is called the lag phase. Whenever you guys take your sample and you put it into a sterile media and then we put it in the incubator to grow, it starts out with that lag phase. It's just adapting to its environment. It's saying, oh, it looks like there's nutrients here. Let's start making our genes to start building again. But it takes a while for the cells to react to that. It's sort of like if you had been starving for a while, you've been working at home, and you finally realize, oh, it's 6 p.m. I haven't eaten all day. Then you probably have to go and cook your food first. Think of that as like your lag phase, okay? So they have to deal with, that change of their environment before that they can start multiplying. Then we start multiplying, it's exponential growth. 1 becomes 2, 2 to 4, and so on and so forth. All right, so we're doubling. That's exponential growth phase. We also call that the log phase because the log phase is talking about logarithmic, same thing as exponential. Okay, so lag phase, then log or exponential. Then the stationary phase. This is where we don't need to multiply as much. We're maybe running out of nutrients. We can't sustain exponential growth. So we'll only divide where we can, and then we'll stop growing, but we're also growing and dying at the same rate. So once we have plateaued, that's the stationary phase. And once we have run out of so many nutrients and our cells are getting old enough and we can't sustain life anymore, once death starts being the prominent thing, then we'll start having that decline in the population, and that's the death phase. So the 4 phases: lag phase, sort of a prep phase, log or exponential phase, exponential growth. Lots of nutrients, we're happy. Stationary phase, we've reached this level of equilibrium, okay? And then the death phase, death is definitely outweighing, and eventually all of them will die. All right. So here's some more explanation on that if you want to visit it. So here's our first question. I think it's 3 questions for this one. There'll be 2 for the next set. So chemo heterotroph. Logan, where's Logan at? Okay, do your best injury on this one. Chemo heterotroph.
Microbes that gain it.
Yes, so they'll get their energy from the chemo part, making the chemicals, and then the heterotroph talks about the carbon source is from the organic molecules from the things that they eat. There's both things going on there, energy and carbon. All right, second question is mine. What would happen if you put a cell into pure water? Well, only the water can cross, right? So there's more salt in our cells than there is in pure water. So we're going to try to water down our insides. So it's going to swell up those cells. So that's hypertonic environment outside, causing the cell to swell. So this is Heather. Where's Heather at? All right, Heather, why does active transport require energy?
Because it's against the concentration of the.
Exactly. So that's why. So anytime you're fighting nature like that, you're going to need to input energy. All right, so we're moving on to our last one, which is chapter 25, microbial ecology and environmental microbes. All right, so this is basically discussing microbes in their natural habitat. First, which this one's going to be a short one, man. I'll keep them short and sweet. All right. Dealing with our elements and how those elements that we introduced in chapter 9 are going to be cycled in our environment. Let's talk about carbon first. Carbon, obviously, in all biomolecules. How is carbon cycled? Well, we already kind of talked about this a little bit. Carbon dioxide that you exhale will be fixed into place by things like plants. So our photoautotrophs. Those autotrophs are going to fix that carbon dioxide into an organic version. And that can be used by heterotrophs, who will then exhale out carbon dioxide. That can then be used again by the autotrophs and cycled that way. But we also have input from our environment, from things like, what is this called? Combustion. So natural combustion from volcanoes or combustion from industry or whatever. And then the carbon dioxide in the atmosphere can be fixed by our autotrophs through photosynthesis. And then our heterotrophs and our saprobes can release it as carbon dioxide through respiration. This is an obvious one. It's not too complicated for our carbon cycle here. If you know, then you know, like we've been watching this pretty much since the Industrial Revolution. The increase in carbon dioxide from our environment absolutely is created by us. There's no denying this at this point, and the fact that it has contributed to global warming, there is no denying this. All science has pointed to the fact that global warming is caused by the increased carbon dioxide rates, and that we have been major contributors to this. Global warming is real. It is, I'm sorry, it is irrefutable at this point. And if you look at the charts at the increase of global temperature, it is real. Now, how detrimental that will end up being and how much we can recover from that, that is still up for debate, of course, and what can be done about it, still up for debate. Most experts will tell you there's not much we can do about it at this point. It's gotten so out of hand that we're not really sure we can do anything. But I don't think that's true. I think it's just going to be a matter of finding the right science to maybe deal with that effect, right? Methane is actually a bigger contributor to the greenhouse gases than carbon dioxide is. So those cows farting in our pastures here in the USA was the #1 producers of beef, right? And all these large pasture areas, all these farting cows releasing their methane from their gut bacteria out into the atmosphere is actually contributing more to greenhouse effects than like just their carbon dioxide that they exhale. So that's interesting, I guess. Whatever. Moving on to nitrogen. We know we need nitrogen for our amino acids. Amino means the NH2 group in our amino acids. So we know there's nitrogen in those, but we also know we have nitrogenous bases in our DNA and our RNA. So you absolutely need nitrogen. As it turns out, plants cannot get their nitrogen from the environment. They rely, like we rely on things like plants to make our organic molecules. As it turns out, plants rely on bacteria to make ammonia in a form that they can use. So without bacteria in the soil, plants can't get the nitrogens that they need. And they need nitrogens to build their cell DNA and their proteins, right? So as it turns out, global warming has contributed significantly to the increase in temperature to the point that now bacteria are unable to grow in soils due to the higher temperatures of the soils. And now the plants can't grow in the soils because there's not any nitrogen available to them from the bacteria in the soils, which is why places like Sri Lanka in our tropical environments are actually going into extreme risks of starvation because they can no longer grow their food because of the global warming effects. When we're talking about global warming having an effect, if you haven't seen the movie Soylent Green, shit's real, okay? It's coming. Like pretty soon, if the temperature keeps rising the way that it does, bacteria won't be able to survive in our soil and we will run out of food while the population continues to explode the way that it has. And then suddenly you're going to be eating people, which is what Soylent Green is about. And if can't get you enough food, they start feeding people the dead people.
You said soil?
Soylent. Oh, soil.
Soylent Green?
Soylent Green. Soylent. Soylent Green.
That's the name of it.
Yeah, it's old. It's like Charlton Heston. Okay. So if you haven't seen it, look it up at least, like read about it. But it's basically like population control has gotten out of hand. Anybody watch Snowpiercer? If you ever watch Snowpiercer, you know the people that are like in the back of the train that they get like the nutrient bars or whatever, and everybody's like, oh, these ones are tasty, and it ends up it's just ground up cockroaches. It's sort of like that concept, okay? Yeah, you guys, yeah. So I don't need you to know what nitrogen fixation versus ammonification versus nitrification. These are all different processes in the nitrogen cycle. Know just that they are involved in the nitrogen cycle. You don't need to know what happens in each one, okay? But know they are steps in the nitrogen cycle. So this is where we're talking about the microbes in the soil and that are embedded in the roots of our plants, that those microbes are converting it into usable nitrogen for the plants. All right. Water on the earth. So this is breaking down all of the water on the earth. Okay. The dark blue is salinated water. So that means it has salt content. That's 97% of the water on the earth. Of the 3% that's fresh water, we have the ice caps and glaciers making up most of that. That's what this big chunk is here. This here is like groundwater. Okay. In here, we have this kind of second area here that gets broken up top up here. This is going to be our other sources. And then we have our surface water here. So from our other sources, this will include our lakes and our rivers and our swamps. Yeah. And so this is basically just breaking down our fresh water that would be a part of that source. All right. So it's a very small proportion of it, basically, that would be usable as far as that. Big problem with our aquatic environments, whether it's salinated or fresh, is the plastosphere. So basically the fact that plastic cannot be broken down by microbes. And so it's just collecting in our atmosphere. And if you haven't heard about the issues with microplastics, then I don't know what rock you've been living under. But microplastic, it says 88% of the ocean's surface is coated with microplastic particles. And I'm sure you've been learning all about how it's in your food and in your drinks and everything like that. There literally are microscopic plastic molecules that you cannot get rid of once they've been in your body. Like they're permanent. They're there forever. So we can find it in bodily fluids, and so in your saliva, in your urine, in your feces and semen, and all of these bodily fluids, you're just excreting microplastics that you're introducing in your body. 88% of the ocean surface, 88%, y'all. 12% of it doesn't have plastic. 12% of it doesn't have plastic. We have islands of plastic literally larger than Texas that are floating out in the ocean right now. And those are the macro plastic islands, blocking all the sunlight underneath it so that organisms like plankton that are depending on sunlight for energy and things like that are no longer getting sun to them. And then all of the micro ecosystems underneath that are affected because there's not plankton available there, et cetera, et cetera, et cetera. Massively huge detriment to our planet. And there is work being done on this, but there needs to be more. Because obviously 88% is unacceptable. Yes.
So do you drink out of plastic cups?
Oh, absolutely. Absolutely. I'm conscious, but probably like all of us, right? Like we drink out of the plastic bottle and we're like, I'm that asshole today. And like, but our society has basically made it so that it's almost impossible to avoid the use of plastics anymore. If you go to areas like California, they're way more conscientious about it than they are like here in Oklahoma. But I mean, I don't know. All you can do is be conscious about it and try to change how you live your life. I mean, even me when I go to Aldi, like I always forget to bring my bag, so I end up using like the paper bags, and that to me feels wasteful, even though it breaks down. I don't know, just stuff like that. Or I don't buy like bottled water, for example. I have an RO system at home, and I have a filtered system that I get filtered water from that. So I don't add to waste with that. But I believe me when I tell you I add plenty of waste. I'm not any. Yeah. See, stuff like that. Like, I'm not judging. As long as we're aware of it, that's the step in the right direction. And then hopefully you would hope that society would help us make that an easier transition to not using as much plastics. But man, the 80s and the 90s have really set us up for failure here now that we're aware of the way that life really works.
Dump their trash into
The ocean, and that's what they do with it. Yeah.
I lived in Hawaii for 7 years. They're very like
conscious of the ecosystem there. Yeah. Yes.
You can't even park on the grass.
Right. Yeah. And that's why things are so expensive there too, is because they only allow certain things into their environment because that could affect their ecosystem as well. So anyways, moving on to drinking water. If you want to go from lake water to water that you drink at your tap, there's steps that we go through. So most of us know that our water is coming from places like Overholser and Hefner. Like if you live in Oklahoma City, your water probably comes from Lake Hefner, right? And I don't know if you know this, but Overholser is a feeder lake into Lake Hefner. And it's part of the filtration process for Lake Hefner. So that's actually the North Canadian River feeds through Lake Overholser, and it sits there to allow things to sediment out. And then it travels through the Stinchcombe Wildlife Preserve, which helps to filter it even further, and then can feed into Lake Overholser. And that's part of how Overholser gets water. If you didn't know this, like this is probably like, oh, okay, these things are actually for a reason. Yes, they are. And actually most of Oklahoma's lakes, if not very, very great majority of them, are man-made lakes that are for this purpose, to provide drinking water and watering for agriculture. All right, so what happens with it is we go from our raw water, from our lakes and whatever, and we're going to sediment that and coagulate anything that we can from that into solids that can be settled down at the bottom. Then we'll take the water off the top of what's actually settled down as sedimentation and filter that, usually through sand, through activated carbon and things like that, charcoal filters. And then we will disinfect it by introducing things like chlorine or UV light to kill off that stuff. And then that is what is distributed to your homes. All right. If we're talking about wastewater treatment, it's another other story. So in other parts of the world where they poop and pee and wash their bodies is also where they drink their drinking water from. And so this is where we see that heavy increase in our parasitic infections in these areas because they use wastewater to wash with and things like that. And you also have toxins and other things like that. So how do we treat wastewater? So wastewater here, I'm talking about you have flushed your toilet, you've gone poo-poo or pee-pee. And now that wastewater has to go somewhere because it doesn't just get stored somewhere, right? We're all aware of that, right? It gets treated. And then when it gets treated, where does it go? It goes back into the environment. So we have to treat it in a way to make it friendly to our environment. So how do we do that? First stage is the primary stage. We're separating out large matter, things like plastic or anything that is actual, like, let's say large, let's just say large matter, okay? If it's a physical solid, it's a physical object, separating that out, okay? Now that we've separated out physically separatable things, next we'll go into the secondary stage where we're going to degrade things with bacteria, algae, and protozoa. They're going to break down what's left from removing our actual like siftable out items. This is going to create sludge. So basically waste from these guys that we can separate out sludge. It is what it sounds like, but it's basically like waste product that didn't get physically separated out and is still within that mess. And so that we can break down from these biodegradation, as well as separate out whatever's left as that waste. And we can use that for things like fertilizer and compost and things like that. All right, and then we have the filtration and chlorination. This is the same as with our normal drinking water, filtration and chlorination and UV treatment. And then that reclaimed water that we got, we can use for watering golf courses. We can use it for watering parks. We can use this for releasing into our larger bodies of water, like our rivers and our lakes. And then, yeah, it will get retreated again before you drink it. But yes, absolutely, you might be drinking your poop and pee water, but it's going to be treated through wastewater treatment and then again through your drinking water treatment. Okay.
Like you saw is the biggest wastewater dump in Oklahoma
Mhm.
and Texas.
Yeah.
Texas will come up and go to Lake Fall to dump. That and the Grand Canadian River up in Oklahoma City.
So we have wastewater surveillance. So you want to make sure that the water that we're releasing into our environment doesn't have viruses or bacteria or whatever in it, and that the water that we're treating for you drink and whatever also doesn't have these things. So we need to be able to monitor for these things. If it is clean and drinkable, if it's free of anything visible, like it looks clear, it tastes clear, and doesn't have actually active pathogens in it, it might not be 100% pure water, but it's otherwise drinkable, safe, that's potable. It's not potable, it's potable. Okay? Potable water is clean and you can drink it, it's free of pathogens. Free of pathogens is the key there. Okay, potable water, free of pathogens. So we want to be sure that we don't get incorporated in any of our water that we're releasing into our environment or that you are drinking from your tap with things like Giardia, Cryptosporidium, Campylobacter, Salmonella, any of this stuff. We want to be sure we're monitoring for that. Well, when you're treating your water and you want to be sure that it's safe for people, you don't always want to wait until you see those bad guys. So what we do instead is survey for things that aren't as bad. So normal E. coli, for example, you won't get sick from just normal intestinal E. coli typically, or other bacteria that are just normal in your gut, like Citrobacter, Klebsiella, and Enterobacter. These are normal gut bacteria that don't normally make you sick. They're just in your gut. But if we can see that in our samples, the bad guys could be there. So we're not going to wait till we see the bad guys. We're going to use these indicator bacteria to look out for them. So these indicator bacteria are what we call coliforms or fecal coliforms. Coliforms are basically our gut bacteria that if they had made it through the filtration process, the bad guys could potentially make it through. Okay, so those are our indicator, our coliform indicators. So basically what we would do is take your tap water, filter it through a filter. It captures all the bacteria on that filter. I put that filter onto a agar plate and then see if anything grows. There is a limit for drinking water and then for wastewater, for like gray water stuff. So there's limits that are allowable depending on how you use the water. Obviously, it's the absolute want zero wherever you can get it, but there's allowable amounts of coliforms for these guys. So that's the membrane filter method. This is basically a picture of how that would work. Filter out your water, then what collects on the membrane, you put onto an agar plate, and then you will count these guys. And the Environmental Protection Agency, or the EPA, is what determines what levels are considered effective and healthy. If you live in Bethany, bank on having that nasty water. Man, they have more boil notices than any other area in the Oklahoma City metro. Bethany, anybody live in Bethany? No? I live in Bethany. That's why I have RO water. Like, that's why I have a reverse osmosis system in my house, because that water is nasty. They don't always get it from Oklahoma City. They have their own water. So they like mix it with Oklahoma City water. And like their water source is disgusting. Like it's like one of the absolute worst in the whole nation. So it's bad. So, yeah.
Mine runs off of well water,
Mhm.
so it's salty.
Yeah, so that can matter too. So you do have to get well water tested just to make sure that you're not leaching anything in there, but that can be better or worse. It just depends. Yes.
So for Bethany, did they get it out of,
I don't know.
because that was kind of known for having people drop bodies and all.
Yeah. Yeah. Like I said, I live right by Overholser, like almost on the lake. And so I'm well aware of Overholser. Usually when we do the pond water experiment, I usually get my water from Overholser because I just live there. So like it's whatever, but it's not nice water. Yeah. All right. Moving on. I actually got water from Overholser. One year I went to like all different like water areas in Oklahoma City Metro. I went to Overholser and Stanley Draper and Arcadia, and I had the absolute most microbes, for better or for worse, I don't know, from Arcadia. So, and that's the swimming lake, you know? And I had nothing in Stanley Draper, but I think that also speaks to how gross Stanley Draper is that it's so gross that things won't even grow in it. So, for what it's worth. All right. So we obviously know how food plays a part in how bacteria play a part with our food. So I'm not going to go over really this too much, but we have yeast that can add flavor as well as air pockets made from fermentation in our bread and in our beer. And they can also make alcohol for us. One health is the concept that all living things on this earth kind of work together to create the overall health of the planet and all things on the planet. That's essentially what One Health is. So all health on all life is connected so that we can maintain the health of organisms other than humans, that it would contribute to the health of humans and vice versa. And then effects of climate change, whether it's, allergen levels, weather extremes, rainfall, and mosquito-borne diseases. If you don't know about the effects of mosquito-borne diseases, we're having increase of things like dengue coming out here in the United States, which was originally considered to be a tropical disease, but we have it now here in the US because of climate changes leading to increased mosquito populations and things like that.